pyruvate. Remember, and this cycle for every sugar, for every glucose, this goes twice, right? Because you break the glucose into three carbons. The pyruvate on the way in loses a carbon, so we got the 2 left. That carbon's lost to CO2, and now you've lost the other 2 carbons. So essentially, when you're doing your bookkeeping, that 3-carbon fragment is gone back in the atmosphere. Okay? And you've already extracted your total energy yield so far is high energy electrons in these activated carriers. Okay? Now we're going to convert, so that converts alpha-ketoglutarate. We lost a carbon, now we're down to 4 carbons. We got succinate, we got this product here. Okay? What happens now, this is interesting. Let me talk about this enzyme right here before we go forward. Alpha-ketoglutarate dehydrogenase. Look at what it does. It does 3 things. It decarboxylates, it oxidizes this fragment to strip away electrons, and it allows coenzyme A to re-link up. What other enzyme does all 3 of those things? So alpha-ketoglutarate dehydrogenase is really almost the same enzyme. It's doing the same 3, just using it on a different substrate. So it's not the exact same enzyme, but it performs the same 3 tasks. It removes it, decarboxylates this to convert from 5 to 4 carbons. It oxidizes the product to remove electrons, and it attaches it to COA. Okay? The energy for this attachment coming from the oxidation. So now we've got succinyl coenzyme A. Okay? The next reaction is now going to hydrolyze off coenzyme A to release a bunch of energy, break that high energy bond. Okay? That energy is going to be used, the hydrolysis of coenzyme A is going to be, that energy is sufficient to attach an inorganic phosphate to GDP, converted to GTP, which is then used to make ATP. Okay, so here's our, this is substrate phosphorylation, right? Okay, remember we talked about, go back to the previous slide. If I can find it, I have this slide here. Substrate level phosphorylation, you have a direct transfer of a high energy phosphate to a target, in this case ADP. So this, just like we talked about in glycolysis, this is the same type of event. Where was I? Here I am. Substrate level phosphorylation. Okay. Phosphate energized by the hydrolysis of coenzyme A attached to GDP, making high energy GTP, and the phosphate is transferred here to substrate level phosphorylation. Okay, so there's some energy payoff. Remember, this cycle is spinning twice for every glucose molecule, so you're going to have 2 of those. So you're up to 1, 2 per acetyl, but 4 per sugar. You got now 1 of these per acetyl, but 2 for the glucose, right? Because you have to double this for every glucose molecule, right? Okay. Okay, and then we're going to have, this is now going to be converted, so all this hydrolyzing coenzyme A, using that energy to phosphorylate GDP, and then ATP. Your product is this 4-carbon sugar here called succinate, or succinic acid. Then you're going to, the next enzyme is a dehydrogenase, another redox reaction. Now in this case, the electrons are stripped away to a different type of activated carrier, FAD. And it's going to produce FADH2. This is another RNA-based activated carrier. Okay? And this, we have to talk about this enzyme right here. This is an interesting enzyme, succinate dehydrogenase. Don't say succinate in a bar, you might get hit. I was talking about the Krebs cycle, not you. Have you ever been in an English pub? There's a guy sitting with a girl. The guy's drunk. He goes, "You look at me, chick?" I go, "No." "Oh, you're not looking at me, chick." It's like, you don't think she's too good looking or she's not. Either way, we're fighting. I know. Okay, let's just go. God save the queen. Okay. This enzyme is the only enzyme in the Krebs cycle that's not dissolved in the matrix fluid. It's actually bound to the inner membrane. It's on the matrix side of the inner membrane. Because it's also going to be part of the electron transport chain, as we'll see. It has kind of a dual function here. We'll see that when we get to it, okay? Okay. So now we've gotten this far. Here's our energy yield so far. Two high energy electrons here, two high energy electrons here, two high energy electrons here, and one ATP per acetyl group. Of course, for glucose, you've got double that. Okay? And now the rest of the chemistry is really to regenerate the starting substrate. That's why it's called a cycle. Okay? So once we oxidize succinate to fumarate, then water helps us to rearrange the molecules added. Our last redox reaction, notice it's malate dehydrogenase. So whenever you see dehydrogenase, you know we're talking about removing electrons and hydrogens. It's a redox reaction. You're harvesting energy. That's what dehydrogenases do. They're harvesting high energy electrons. So now we per acetyl, we've got now 1, 2, 3 NADH, 1 FADH, 1 ATP, and we're generating oxaloacetate. Now the cycle can happen again. Okay? And really, this is the center. The Krebs cycle, the citric acid cycle, is the center. Okay? It is the focus of energy metabolism in eukaryotes, I should say in aerobic organisms. Okay? Oops, I need to do that side. That's wrong button. This is really the centerpiece of aerobic respiration. Okay? Because the energy in pyruvate, before it's oxidized acetyl, all the energy for excess ATP beyond glycolysis is here. It's in the form of high energy electrons. They have to be stripped away, and that's what this process does. It dismantles the remnants of this sugar fragment and harvests all the high energy electrons it can get. It does make a little ATP through substrate level phosphorylation, but the main harvest is the electrons. And at the end of this, the sugar is all lost back as CO2. Remember, that's carbon's lowest energy state. Okay? Now, oxygen is not directly involved, but if there's no oxygen available, this pathway shuts down. Okay? Because these activated carriers are in limited supply. Okay? And when you're reduced with electrons as NADH or FADH2, you can't absorb more electrons and keep the cycle going unless you can get rid of those electrons. Move to an electron transport chain, which requires oxygen to kind of suck the electrons energetically downhill. No oxygen, these remain reduced, cycle stops. Okay? So although oxygen is not directly involved, it's required for this chemistry to run. Does that make sense? And why won't it run? I'll ask you what I just told you. Can you tell me why it won't run if there's no oxygen present? What stops this cycle from working in the absence of oxygen? Yes, ultimately. And if the activated carriers don't lose their electrons, they can't be the recipient of these redox reactions, the whole thing stops. Right? Does that make sense? So, although it's not directly involved, without oxygen, this pathway has to stop. Because these have to be recycled back to their oxidized form to keep the cycle going. And they deliver the electrons to the electron transport chain, whose final oxygen trap is oxygen. There's no oxygen to pull them downhill. They're not going to go anywhere. They're going to stay here. And then these reactions cannot occur. So how many times does the Krebs cycle happen per glucose? Per glucose, how many times does this cycle run? All right, here we go. All right? Okay. Ooh, another nasty slide from your textbook. Okay. Now we're ready for the big payoff of A to B production. The electron transport chain, the chemiosmotic production of ATP. Being an electron transport chain that builds a proton gradient. Okay? So again, it's very freaking complex. But it's not my fault. Okay, there's another. These are good figures in your book, but you try to show them on the screen and can't read anything. But so I'll show you some other versions from other textbooks here. But let's look at the. So now we're here, and what this transport chain. You see, its membrane is essentially 3 large protein complexes made up of over 40 different proteins. Okay, that serve as a pathway for electrons to flow toward oxygen, energetically downhill. Why is oxygen energetically downhill? Because it's so freaking electronegative. Okay, and it wants to attract electrons. When it grabs them, it doesn't want to let them go. Okay, so it's like a suction force. You want to think of it like that. Right? So we've got 3 main complexes. They're numbered 1, 3, and 4. Complex 2 is here. And actually, that succinic dehydrogenase is part of complex 2. They talked about that enzyme in the Krebs cycle. We'll revisit that. But they're embedded in the inner membrane. This just shows you one set, but there are millions of these in the inner membrane that's highly enfolded inside the mitochondria. Okay? And the electrons are delivered to the complex via the activated carriers. Remember, this is the matrix here. Here's the cytoplasm. Here's the intermembrane compartment. Here's the matrix where the Krebs cycle just produced these activated carriers carrying high-energy electrons. They each carry 2 high-energy electrons. And they deliver those electrons to the electron transport chain. Okay? And then they are oxidized in the process so they can now participate in extracting more electrons from the Krebs cycle. Okay? Now, as the electrons are shown in blue, their path in blue. Okay? We'll talk about some of the components of this system, how it works. And then the red arrows, you can see the red arrows. This is the movement of protons from the